So population genetics 2. We've gone over some basic Hardy-Weinberg equilibrium. Now we're going to go into applications of Hardy-Weinberg equilibrium and exceptions to that. We're going to look at specific fitness rates. We're going to look at selection, drift, non-random mating, and migration and the effects thereof. We'll also get into what's called neutral evolution, non-Darwinian evolution. So it's Hardy-Weinberg. What's the sum of the allelic frequencies in a population? What does it all have to equal? 1. Whenever you have a p plus q, you get 1. P squared plus 2pq plus q squared, you equal 1. So Hardy-Weinberg, what it tells you is the frequency of the genotypes in the next generation. For Hardy-Weinberg to hold true, for it to be predicative, you need to have no mutation, no migration, no genetic drift, no natural selection, and, this is going to sound weird, no non-random mating. Now, what happens when Hardy-Weinberg does not equal 1? What do you get? Evolution. You're right. Mutation could be one reason for it, though. So we're going to start off just reviewing some basic Hardy-Weinberg. All right, so this one again, in a given population, only the A and B alleles are present in the ABO system. There are no individuals with type O blood, so everybody with type O blood has been wiped out. The type O allele apparently became deadly at some point and killed everybody who had it. So we only have A and B present. So you automatically know what the genotypes are. Big A is type A. Big B is type B. Big B is type AB. We know that 200 people have type A blood, AA. 75 have type AB blood, and 25 have type B blood, so BB. What are the allelic frequencies? So we have our totals of 200 type A, 75 AB, 25 type B, leading to a grand total of 300 individuals in the population. To get type A, how many type A alleles are there? 200 plus 200 plus 75. So 475 over a grand total of 600. What? Was I wrong? Right? Oh, I forgot to do, so 300 times 2 gives you 6. So 475 over 600. And then for type B, you could do it one of two ways. You could count up all the type Bs. 75 plus 25 plus 25, which is 125 over 600, or subtract 1 minus 475 over 600, which gives you 125 over 600 for Q. The end. Sound good? Questions? Moving on. Again, basic Hardy-Weinberg problem. Here we've got PTC tasting. Now, I know some of you guys remember distinctly the PTC taste. It's not good. PTC tasting is due to a single dominant allele, T. Big T means you're a taster. You sampled 215 individuals in biology and determined that 150 can detect it. So 150 of them can taste, that is, they have the big T allele. 65 cannot taste. They do not have the big T allele. Calculate all the potential frequencies. Tell me all about P and Q and the homozygous dominant and the heterozygous. What are the frequencies? So what's the first thing you need to determine? The most important thing for determining this. Well, you know that there are two alleles. You do need to determine the allelic frequency, P and Q. How can you determine P or Q? Because if you have one, you have the other. Say it again. P plus Q has to equal 1. What are you given here? You're given the number of dominant individuals. So there are 150 dominant individuals, and then there's 65 recessive individuals. So that's right, specific over the total. So you get 65, say, hypothetically, over 215 gives you what value? What does that equal out to? What? Little t? Yes. Q squared. So if that's equal to q squared, what's q? That's right, square root of 65 over 215. So then what's p? That is correct. So far, so good. So what do you get for the homozygous dominant frequency? P squared. So P squared is the homozygous dominant, which is going to be 1 minus the square root of 65 over 215 squared. And then what's going to be the heterozygous frequency? So 1 minus the square root of 65 over 215. Okay, that's one of the things you need. Times the square root of 65 over 215. And one more thing you need. Not an exponent. 2. Because it's 2pq. Pretty close, though. All right, so we figured it out. We've got our q, square root of 65 over 215. P, 1 minus the square root of 65 over 215. P squared, 1 minus the square root of 65 over 215 squared. And 2pq, which is 2 times 1 minus the square root of 65 over 215 times the square root of 65 over 215. Those are all the possible frequencies. One more problem. It's all about the albino squirrel. Albinism is a recessive trait. If a population has an albinism rate of 4% and the population is in Hardy-Weinberg equilibrium, what would be the expected genotypic ratios of the rest of the population? Give me also the phenotypic ratio. Should be really easy at this point for you guys. So albinism is a recessive trait. Population in the population is at 4%. 0.04. So what does 0.04 equal? If it's the recessive genotypic frequency. So Q squared. Not Q2, Q squared. All right, so that means that Q has to equal the square root of 0.04, which is going to be equal to 0.2. Because that's how math works. All right, and if Q is 0.2, what does P equal? 0.8. So what is the frequency of the heterozygote? 2pq. 2 times 0.2 times 0.8, which is 0.32. Then we have p squared, which is 0.8 squared, which is 0.64. So you have an albinism rate of 4%, a normal weight rate of 96%, homozygous dominant 64%, heterozygous at 32%. The allelic frequency of P is 0.8 and of Q is 0.2. Feel a little better on that? All right, new idea. New topic. New topic, same as the old topic, natural selection. We're going through a lot of those Hardy-Weinberg exceptions again, just in a little bit more detail. Natural selection is the process by which we get a beneficial heritable trait. It must be heritable. That will increase the allelic frequency in successive generations. If it's not heritable, if it's just a trait that pops up and you can't pass it on, that's not natural selection. You have to be able to pass on the trait. For instance, if you ended up with a mutation, and it's a somatic mutation, a mutation in your body cells that is not passed on to your offspring, even if it helps you live, It's not natural selection. Selection needs successive generations. This, of course, is one of my favorite pictures. I love the picture of the, you can't see it all that well up there, but some guys decided they needed power in their pool, so they floated a power strip on some flip-flops. Love that. Basically, adaptation is going to go in two ways. Natural selection promotes adaptation. If you have a new allele that comes into a population, in this case, we've got primarily green bugs, beetles. There are some brown beetles too, but not many. If the crows prefer the green beetle, they will only eat those green beetles, leading the brown beetles to be able to reproduce. Over time, the allelic frequency of the brown beetle increases as the green beetle decreases, until finally there aren't very many green beetles. Now, this doesn't show the whole story. This shows what's happening on the beetle side. What's going to happen to these birds once they run out of green beetles? Well, they start to die. Yeah, but until you get a crow that can or prefers to eat the brown beetle. Once you got that crow that prefers to eat the brown beetle population decreases, and you start to get a balancing effect. We're going to see this motif over and over and over again. It's called the Red Queen hypothesis. Anybody, you guys ever read Alice in Wonderland, Lewis Carroll? There's this scene in there where the Red Queen tells Alice she has to run as fast as she can just to stay in the same spot. That's evolution. Everything is adapting very quickly to the world around it. Predator and prey are both mutually adapting at the exact same time. Some changes will be beneficial, some deleterious. In the end, it leads to a net result of zero. Nothing, there's a balancing act. Now we can measure how well an organism survives, sort of so we can tell its rate of survivability. It's called its fitness, the measurement of reproductive success. Now we can measure reproductive success in a whole lot of different ways. You can measure reproductive success by the, the number of eggs, the number of offspring that are born, the number of offspring that survive to adulthood. Do you guys know this family? They were on the Learning Channel for a while. I forget their names. The Duggars? Yeah, they've like a gajillion kids, 21, 22, 23, whatever. They just kept churning out kids. That was like their thing. By any measure of biological success, they're winning. I mean, they've passed their genes on to the next generation. Those kids, if they go on to have big families, will pass their genes on to the next generation. And these will spread like wildfire. If you have genes that allow you to have a higher level of reproductive success, you win as far as biology goes. You have two kids, you've managed to pass on your genes exactly, you've managed to reproduce or replicate pretty much, not exactly, but your gene structure and your mate's gene structure in the next generation. More than that, you've increased your genes in the next generation, the frequency of your genes. Less than that, you've decreased them. So we're looking at different measurements of fitness. So modern natural selection, modern evolutionary theory is based entirely on molecular biology. You get a new mutation that creates a new allele. It's not based on simple, look at the structures, what do they do? You change the allele. That might alter the amino acid sequence. Remember, central dogma of molecular biology. Transcription, you're going from DNA to RNA, translation from RNA to a polypeptide. That can alter the function of the protein. You change one single nucleotide, you've changed the codon it's in, you change the codon, you might change the amino acid, you change the amino acid, you change the relationship between that amino acid and the other ones, causing secondary structures not to form properly, leads to tertiary structures not forming properly, leading to a misfolded protein. Sometimes those misfolded proteins will give you a benefit. Sometimes they're going to take away. Sometimes they help you reproduce. Other times they stop you from reproducing. So some of the alleles enhance your fitness compared to others. Here we've got two types of fish, a light color form and a dark color form. These are the same species. What you'll tend to notice is the light color form does really, really well with light sandy banks. The dark color form does really well living in creeks that were at one point filled with a lava flow, because lava flow is dark. These guys, the dark ones, have a reproductive advantage in one setting. Their fitness is increased. The light ones in that same lava flow aren't going to, not in the lava flow, they would all die there. In the river that had the lava flow are different. Individuals with more beneficial alleles are more likely to survive, and they're more likely to contribute their genes to the next generation, whether you're a protozoan, a shark, or a turtle. Doesn't matter. By the way, these are some of the pictures that got me in trouble with the library when I was rating these lectures. Apparently, writing turtle sex into Google search raises a flag. Didn't know that at the time. So these are more likely to contribute their alleles to the next generation. By the way, you have to appreciate how dedicated sharks are to the act of mating. They bite onto each other. They hold, like literally, a shark will bite another shark, hold onto its fin, and then ram their face into the ground. That's dedication. So you're increasing allelic frequency of the beneficial allele, decreasing the detrimental alleles, leading to a net increase in the fitness of the overall population. So we can measure fitness in lots of different ways. One way, count the eggs. How many eggs do you lay? How many eggs do you have in your body at any given point? Another way, how many babies are born? Third way, how many copulation events do you have? Any of those can work as a measure of fitness. Successful copulation events. My graduate work was with fruit flies. I was looking at speciation and evolutionary mechanisms in Drosophila ananassi. Now, if you guys have seen fruit flies before, right? You've seen them in your house and you hate them. You know what I'm talking about? All right, fruit fly. Imagine a fruit fly that's about a quarter of the size of the ones that live in your house, because you've got melanogaster in your house. They're pretty big. Really tiny guys. I had to measure the fitness of those guys. The way I had to measure fitness was two ways. One, dissect them, count the number of eggs they had. Literally, dissecting a fruit fly. It took a very sharp needle, knocked them out on the microscope, looking under the scope, slice open their belly with a needle, and then try and pick out the eggs from the rest of the assorted bug guts. Tricky. The other way I did it was I literally had to sit there and watch them as they mated. I fed them different colored foods so that some of their bellies were green and some of their bellies were red. They'd fly around in the cage. I would sit there like the really sick guy I was at the time, apparently, like this, and watch a cage for 3 hours, looking for any flies that mated with each other. And then I had, do you guys know what a pipette tip is? Okay, so it's a little plastic cone, I guess, teeny tiny, and a tube. And I put the tube in my mouth, and I put the pipette tip in there, and I'd suck the mated pair out and put them in the tube all by themselves. So I'd be able to count how many mating events there were. Very quickly, I learned it was very important to put cotton in between my mouth and the fly, because you suck too hard, and that fly goes right down your throat. That was never fun. But these are fine measures of fitness. How many eggs? How many mating events? How many offspring? Any of those will work. Eggs, copulation events, offspring. Which brings us to a whole new set of mathematical equations. Don't worry, though. These are way easier than the last ones. W is what's known as the fitness value. It's a relative fitness value. So that means what you're going to do is when you're comparing the fitness of different genotypes, big A, little a, big A, big A, little a, the one that has the highest fitness for whatever you're measuring is given a value of 1. And all of the others are set to that as a standard. So what that means is, I know, that it sounded crazy. Sorry, your brain hurts. Let's say we've got 3 genotypes. Big A, big A. Big A, little a, little a, little a. All right? These guys are going to mate a certain number of times. How many times does big A get to mate successfully? Twice? Wow. How about big A, little a? How about 12 times? Because they're just that much better at it. Little a? 42. Sounds good. It's life. Which of these is the most reproductively successful? Little a. They're really good at mating. Make sense? So you're going to set that equal to 1, which is 42 over 42. All of the rest you set against that same standard. So now it becomes 12 over 42 and 2 over 42. So those are the fitness values, the W values. It gives you a sort of standardized way of evaluating this data. So that's W. Let's work out a W problem. Here we have chickens. We're going to measure their fitness by the number of eggs they lay. Genotype big A produces 12 eggs, big A, little a produces 5 eggs, and little a produces 1 egg. What are the relative fitness values? What is the fitness value of big A? 12 over 12, or 1. What about big A, little a? 5 over 12, good. And little a. 1 over 12. Isn't that so much nicer? Yeah, that's nice. Questions on that? One more. Lizards. This time we've got lizards. What are they doing? 14 lizards with genotype big A, little a produced an offspring, while only 7 lizards with genotype big A, and little a produced offspring. So you get 14 lizards from big A, little a, and 7 from big A, and 7 from little a, little a. So what is the fitness value of big A? 1/2. Fitness value of big A, little a? 1, and little a, 1/2. Let me give you another one. One more. Let's see, we've talked about laying eggs. We've talked about copulation events. What was the last one? How many offspring they produce. So we've got one set that produces an average of, okay, let's say people. Certain genotype of people produces how many offspring? 3. They're just like, I'll have a decent number of kids, 2 to replace me and a spare one. All right? How about the big A, little a? What do they produce? None. They're millennials and destroying the world, apparently. How about little a? 17, because they live around here somewhere. So we've got 3 offspring, 0 offspring, 17. Which is the most reproductively fit? 17. So what's the W value? 17 over 17, which is 1. No, that doesn't matter. No. All that matters is biologically. How about this one? 0 over 17. And this one? 3 over 17. Just that easy. But you're bringing up another point. How many of them would survive to adulthood to reproduce? That's another measure of fitness. Cool part is you as the scientist are the one who decides what measure of fitness you're going to use. So, natural selection. We've been talking about different types of natural selection for a while now. Now we're going to look at the actual impact of those selection events. One type of selection is going to be what's called directional selection. Go figure. It means going in a certain direction. It's where one extreme phenotype is selected for and the other ones are selected against. So here we've got a graph showing directional selection one generation to the next. In the first generation, this sort of dotted line there, the dotted line to the original population. you could see that the mean, I don't know, height or the mean weight, that y variable, that y-axis, is going to be highest at an earlier point along the x-axis, along the independent variable. The second generation, these taller ones or these bigger ones, the ones, whatever trait we're selecting for, height, color, whatever, the ones that are more extreme are selected for. That means these short ones don't really get to mate much. So these guys out of the gene pool, thus the average height gets, or the average weight, or the average whatever quality you're looking at gets higher. If we're just looking at height, it's pretty easy. You can see a shift from shorter people at the mean to taller people at the mean. So that's where selection is moving in a specific direction. And that's usually what you think of when you think of selection. Thinking about the Darwin's finches, you got their beaks, right? Their beaks go from really big to smaller and smaller and smaller and smaller, a certain direction. That can be contrasted with stabilizing selection. When you're undergoing stabilizing selection, the two extremes are, both extremes are going to be selected against. So I guess if we're looking at height, shorter heights are selected against. People just don't want to mate with short people. And taller heights are selected against. I don't know, they keep running into doorways and dying. Whatever. The big thing is you've selected against the extremes, so you end up with a center phenotype that's in much higher population density. You see that middle term a lot more. So that's stabilizing selection. Directional's moving in a certain direction. Stabilizing stabilizes at a mean. Diversifying selection is where both extremes are selected for simultaneously. If we're looking at height here again, I don't know, short people prefer to mate with short people, tall people prefer to mate with tall people. Thus, over time, you start losing the single distribution and start seeing what's called a bimodal distribution, two distinct humps. This will eventually lead to what's called speciation. We'll talk about that shortly. Diversifying selection is a really interesting, it's a very interesting concept. Here we've got beaks. Oh, question? Yeah, sure. It may mean no heterozygosity, but we're not just talking about allelic frequencies here. We're talking about phenotypic effects. We're talking about what it looks like. Exactly. You're getting more and more really short or really tall and just a few that are average. So it's important to note here we are looking at phenotypes. We're not just looking at the genotypes anymore. We're not just looking at allelic frequencies. Now we're looking at sort of a combined effect. All these genes working together to give us this look. Here we got these three moths, right? With diversifying selection, you might have this sort of center color, this average color one. That looks palatable. It looks delicious. But the extreme edges, they mimic poisonous species. So even if this moth is not poisonous, in its normal state, it looks delicious and is eaten a lot. The frequency of that phenotype decreases dramatically. Meanwhile, the two extremes are selected for. They stop predators from eating it. So those phenotypes increase. So what you're going to end up seeing is more and more of the different forms instead of the original median form. It's all about selective pressures. Another type of selection, because there's so many of them, is balancing selection. This is one of my favorites. This one is about the heterozygote. This isn't just looking at the phenotype. This is looking at the alleles. Sometimes a heterozygote will maintain a genetic polymorphism. A heterozygote will keep what would otherwise be a deleterious allele or an allele. It would stop an allele from going to fixation. One example of this was the study that they did with the major histocompatibility complex, the MHC. The major histocompatibility complex is a series of genes that's responsible for creating antibodies. So if you have, I'm just going to sort of make this up, gene, big A, big B, and big C. This might produce a protein that's just a solid line, a squiggle, and a triangle. All right? Gene, little a, little b, and little c, I don't know, makes a dashed line, a squiggle going in the other direction, and an upside down triangle, whatever. These are all different forms, thus they all have different functions. If you were to have big A, then you have a solid line and a dashed line. Two different proteins being created that can do two different things. Oops, my fault. Two solid lines. If you got a big A and a little a, now you've got a solid and a dashed. So here you can only bind with one type of bad thing, one type of bacteria or some such. Here you can bind with two. Little a, you can bind with just one type. So it's better to have that heterozygous one. You can have more forms of your proteins. What they found was, and this is kind of interesting, they had people work out. So they gave them T-shirts, the people put on T-shirts, and then the men and women would work out. And what they ended up finding was no deodorant, anything like that, so it's kind of disgusting. They had these people work out, then they hung up the shirts on a clothesline and had women go through, both women and men, but only the women could tell the difference. Women went through and smelled the men's shirts, and they would say which shirt smelled the best to them. And what they found significantly, very significantly, was that women prefer the smell of men who are heterozygous to them at the MHC, who have the exact opposite allele for them at that locus. Because we still have senses, we still have instincts. It's kind of interesting. Granted, you're not going to encounter that very often in real life because hopefully we use deodorant and such to mask our own normal smells, but it's still there. It's a form of balancing selection. Another form of balancing selection is seen with sickle cell anemia. It's called heterozygote advantage. This is where the heterozygous state, if it's otherwise deleterious, increases the fitness of the organism more than the homozygous normal allele. The best example of this is sickle cell anemia. With sickle cell anemia, if you are homozygous recessive, you have sickle cell anemia and your chances of living decrease dramatically. Your blood cells will pile up against each other, restricting blood flow, and could cause issues with the heart. You have a much lower life expectancy than anybody who's heterozygous or homozygous normal. But if you are homozygous normal and living in sub-Saharan Africa or at this point, in South America, you can end up with malaria. Malaria will drastically decrease your life expectancy. People who are heterozygous do not suffer from sickle cell anemia and, coolly enough, are immune to malaria. So you've got two extremes, not two extremes, but you've got the heterozygote providing a major reproductive advantage. If you're heterozygous, you are living in these sub-Saharan regions or in these tropical regions, you are way more likely to survive because you're not going to get malaria and you're not dying of sickle cell anemia. So that's heterozygote advantage. Sexual selection will occur when one gender or the other chooses their mates based on very, very specific sexual characteristics. If you see something weird in nature, and you look at that and you're like, why does that organism have that particular appendage, or why does it do that? More than likely, it's because of sex. Sexual selection causes the most outrageous features to form, like the peacock's tail. This is the peacock, in case you didn't ever see one. They've got these gigantic tails. It is not conducive to survival. But man, the female peahen loves it. She looks really boring, actually. I think the next slide has a good picture of a peahen. It doesn't. We'll get to a peahen in a second then. The peahen is really, really boring. Peacock, very outrageous because he's trying to attract her attention. He's trying to get chosen. We have two types of sexual selection: intrasexual selection and intersexual selection. Intra is within a sex. Two members of the same sex compete directly for the opportunity to mate. Two male kangaroos fighting it out. Two male elephant seals fighting it out. Two bucks fighting it out. These are two members of the same sex fighting in order maybe to claim territory, maybe to lay claim to a harem of females. So that is intrasexual. That's contrasted with intersexual sexual selection. Intersexual selection. This is where one member of a gender chooses a member of the opposite gender to mate. Over here on the left is the peahen, very small, very brown, very gray, boring. Over here is the peacock, huge, gigantic display. That display puts him at a disadvantage for survival, but an advantage for reproduction. If he is being chased by wolves, a peacock with a shorter tail can escape faster and easier. One with the long tail, not going to make it. But odds are that long-tailed one got to mate beforehand, thus propagating those genes. Here we've got I think these are our knoll lizards. What these lizards will do, they've got this white patch under their throat, and they take a deep breath, and it blows out their throat, and so it turns bright red. You can see the blood vessels through it. And that's to attract mates. Bullfrogs do something similar. They make that sort of that deep bass sound. They can blow out air through this in order to sort of say, look at me, don't you want to mate with me? And then, of course, you got the red-breasted robin. The male is red-breasted, the female, not so much, because that male is trying to attract attention. All of these traits, all of these weird traits, think of the narwhals. You guys know what narwhals are, right? They're like whales with these big horns coming out of them, this tooth growing out of its mouth. Straight forward, 6 feet long. Why is that there? Sex. Because seriously, and you know you looked at that and you're like, it must use it for hunting. Somebody thought that at one point. Goes along spearing fish. That's what a lot of people think about. Not kidding. Ask people what the horn is for in a narwhal, and people are like, yep, it's definitely for fishing. It's not for fishing. Why would that be a horrible idea for a narwhal to use to fish? Yeah, that would be horrible, wouldn't it? It stabs the fish, and the fish is just like just out of reach. Almost to its mouth, but not quite. It's all about tickling the other gender. Or with narwhals, what they do is the males will joust. So these are sexually selected traits. And they might lead to bad things. They might lead to lower survivability. If these guys are showing off, they're going to be much more visible to a potential predator. And the funny part is people look at this like, silly animals, you're all doing stupid things. Look at people. We are constantly doing stupid stuff to mate. All the time. The best place to look at this is in a high school. High school boys are hilarious. If you've never taken up a study of high school boys, do so, because they are funny. Think about it. What do? You guys have been in the lunchroom, right? Have you ever seen a fight in the lunchroom? Okay, what's the first thing that happens in a fight in the lunchroom? You get loud. Are they sitting down? Oh, no, they're standing up. Not only do they stand up, and I swear I've seen the same thing happen here in committee meetings. People stand up and they puff their chest out. They're just like, and they take a deep breath and they make themselves look bigger. It's true. You've seen it. And then what do? They bounce against each other. What's that going to do? Even better, they start pushing each other. You see this like, and those pushes aren't meant to actually hurt each other. It's just to say, this is my territory, not yours. I am here, you are not. You will be gone. Big chest. no idea, but it sounds hilarious. You've all seen these displays. It is straight up harkening back to when we had to deal with this for sexual selection. More importantly, now we got intersexual selection. Now we want somebody to notice us. How do high school guys go trying to get noticed? What do they do to stand out? They're annoying. Okay. Why are they annoying? What do they do? They throw. You have a very interesting. I just don't think they like you. They act out in class. Have you seen the cars they drive? What are the cars like? A lot of, if they can, giant trucks, right? Very loud, deep bass, just like frogs, just like the lizards. Very showy. Behold, I can provide this truck. Mate with me now. Humans are dumb, and we don't realize how dumb we are. We are still falling back on these same intersexual selection and intrasexual selection behaviors. Hopefully it goes away as you age. This sexual selection is going to be balanced by predation. These are guppies. Guppies are all sorts of colors. You guys have seen guppies before, right? You go to the pet store and there are all these different colors. If you haven't, they're like $1 each. Guppies are everywhere. Bright, bright colors, huge tails. The bigger the tail, the brighter the color in guppies, the more likely that male is to mate. Females, they're boring. They just look like normal fish. These are all males. Big, big fins. Big, big colors. You don't see males like this in nature because the fins that are that big slow them down. The colors that are that bright make them more visible. Yes? You would think that, and that's what it turns out. There are very, very few examples of females that are the ones that have to do females are usually the selectors. Males are the ones who are getting selected. The difference is the ones where that's not the case would be like clownfish, where the female will select. What? Seahorses, I believe, are together for life. There are a few animals that actually stick together forever. So do females. No, females have the same push. That's definitely the sort of preconception is men are out to sleep with everything, females are out to tend a few eggs. But it's not the case. It all comes down to females want to have as many offspring as they can, just like males want to have as many offspring as they can. We may disagree with that because we're rational beings, but in general in the wild, it's all about how many organisms can you produce. You win the game of life if you make the most. So, with these guys, though, it's bright, it's shiny, and it's deadly. So you don't see this often. It's balanced. Remember, every single trait is being selected for simultaneously. You're not just looking at one trait at a time. You're looking at the whole organism. Now, we just had that whole lab on genetic drift. Genetic drift is all about the game of chance. You can have fluctuations in allelic frequencies, changes from one generation to the next, based solely on chance. Here we've got two maps. The top genetic drift map is showing the effect of genetic drift in a small population, the lower one in a large population. So top is small population, lower is small, top is small, lower is large. What differences do you see? What's the biggest issue? What do you notice between small and large populations? Yeah, the smaller one's got a lot more variation to it. And you'll also notice that the alleles go to fixation very quickly. You're going to have some up at frequency of 1 and some at frequency of 0 within the first 10 generations. By generation 25, you've got a whole bunch of them sitting fixation. But if you look at this much larger population, the first allele to go to fixation looks like it's around 45 generations out. So large populations, genetic drift isn't as important a factor. The smaller the population, the more important genetic drift becomes in changing allelic frequencies. Which is pretty much exactly what this is showing. So you got 25 stoneflies at the start, 500 stoneflies. With 25, fixation with every single line within 30 generations. With 500 stoneflies, you do not get fixation at all. Likely this will just keep going in perpetuity unless a bunch of random events happen in quick succession. So small population, highly impacted by genetic drift. That population bottleneck is a form of, sort of an extreme form of genetic drift. You're going to take a whole population with a varying set of allelic frequencies, and only a small number of that population will go on to survive or go on to found a new population. This has happened several times that we've recorded. Whales. We almost hunted whales to extinction. We really liked their oil. Then we found oil underground. We're like, oh, who needs whales? We hunted whales down to within, I think it was less, like for some species of whales, almost to extinction, close to maybe 500, 600 left. That means that when the population started to recover, and the whales are recovering, the only alleles that could be in that population were the only ones within that 500. A more extreme example would be walruses. I don't even know. I think we were hunting walruses for their tusks and meat. But walruses we hunted down to 200 individuals. 200 walruses in the entire world. And the only reason we didn't hunt those walruses was because they were on an island we didn't know about at the time. From those 200, the rest of the population's rebounded. Now there's, I think, 60,000, 70,000 walruses out there. But the alleles for those 60,000 or 70,000 all came from those 200. There's no different allele. They're very, very inbred. There's high levels of homozygosity. Lots and lots and lots of homozygous individuals. Another good example is stellar sea cow. It's extinct now. We ate it to death. Which, truthfully, is how a lot of organisms died, is we were hungry. Stellar sea cow was, you guys know what a manatee is? They're about 6 feet long, 7 feet long. Big, fat, underwater. They're like sausages with fins. The stellar sea cow was 35 to 40 feet long, but the manatee. And it lived in very, very shallow water. So the water it lived in was roughly, I don't know, 3 feet deep. And it would come in near shore. It had these little claws on its fins, and it would walk its way in and just eat the grass. That's it. Very gentle creature. Lots of accounts of it just sort of hanging out. When human hunters came up, we just stabbed it to death, and it was just like, oh no. I'm big and fat and can't do anything. And then we pulled it on the land and we ate it. I know. And then they're like, you know what? This is great. We've got all these. We found one island with these things in it. Our ancestors before that hunted them to extinction. But on that one island, they were much smaller than they'd been all over the rest of the world. Because that one small population, that founder population, only had those small alleles, those small genes. Does that make sense? Nowhere else could they repopulate larger genes from. So that's a bottleneck. When you end up with sort of lowered levels of allelic diversity. Yeah. 40 feet long. Okay, so 40 feet long would be, each of these is 2.5 feet. So 1, 2, 3, 4, 5, 6, 8, 9, 10, 11, 12, 15. 16, 17. Okay, so from the door to about here. Yeah. Crawling along the rocks. Isn't that wild? They were also Arctic creatures. They love the cold. So, full disclosure, stellar sea cow is one of my favorite organisms to study. I did a big study on them, and they're always so interesting. When you establish a new population, that's called a founder event. Whether it's, I don't know, redheads showing up in Scotland in a small group and then populating the island with redheads. Or, even more cool, Krakatoa. Have I told you the story of Krakatoa yet? All right, here's the story of Krakatoa. Krakatoa was an island. It was a volcanic island, and it blew up. It was the loudest sound ever recorded in the entire world. Even since then, we have never heard a sound as loud as Krakatoa exploding. It was literally heard halfway around the world. So this island exploded, and nothing was left of it. Literally, island was there one day, gone the next. Underwater, after the volcano went, a new island was forming. Volcanism was occurring, magma was pouring out, and this volcano, the island was building itself up again. And everything was wiped out, down to the bacteria, everything gone. All right, now we have this nice volcanic island made of magma on top in the middle of the ocean. Nothing lives there. Over time, sea spray, salt, sand wash up. We start breaking down, the lava starts to, the rock starts to break down. You start to get some soil buildup again. Bacteria have showed up, you get some fungus, some lichens, life's good. On the currents, the water currents and through the air, grasses landed, seeds landed. So you start to get plants growing. Well, now you have this island devoid of life with a bunch of plants. So seabirds come and land on it, and they start nesting there. Well, between what they eat, their guano, and not guano, their droppings, you end up having a more diverse diversity. Insects show up, things like that. But what was really, really cool was there was a monsoon or a weather event, and a single pregnant Komodo dragon ended up on this island. One single, they traced it back. One Komodo dragon, they think the Komodo dragon ended up hitting this island on driftwood. Okay, one, imagine if you're just out swimming and there's a Komodo dragon floating by. But two, it ends up on this island, filled with birds. It must have been paradise. She lays her eggs, she eats the birds, her children hatch, they eat the birds, they get big and strong and eat more birds. This Komodo dragon population explodes, all based off of one female's eggs. The only other Komodo dragons around are siblings, so they mate back together. And you end up getting a whole lot of homozygosity. The same genes are present over and over and over again. The only genes that are there are the ones that the mother and whatever father she mated with before she got to the island. There are no other possibilities. That's an extreme founder event. So a big consequence of a founder event or population bottleneck is you lose genetic variation. You guys have seen lab mice before, right? If you haven't, they're ugly. Lab mice, I hate them. They're white little creatures. Oftentimes they're either all white or all black. Doesn't matter. The big thing is you started off with one mouse. Okay, two mice. You have to have two mice. They have babies. You take those babies. You cross them back together. You take those offspring. You cross them back together. Over and over and over again. Until you end up with offspring that look very, very similar and are similar genetically. They're almost clones. They're that similar to each other. Why would that be desirable for science? Why? You're right. They are good test subjects. Go ahead. Right. Your variables stay the same. In this case, the variable is those genes. The gene interactions in one are going to be the same gene interactions in the other, and the same gene interactions. So if you're seeing an effect of a drug, you're sure, and that drug's hitting multiple organisms, you're sure it's the drug's issue, not some random combination of genes. So they are almost genetic clones. As far as genetic drift goes, a lot of the variation we see, and we're talking about things like, people talk about DNA fingerprinting, things like that. We're all different. Yes, we are all different, but it's not necessarily in our structural genes. We're not all different in the way we make ribosomes, for instance. That's very highly conserved. If you can't make ribosomes, you die. You don't ever exist. We're different in the polymorphic genes that don't matter, the ones that aren't selected for. These genes are termed neutral. Any gene that is not selected for or is not selected against, there's no selection pressure on it whatsoever, is neutral. What parts of the genome would have absolutely no selective pressure on them? What parts of DNA, what parts of a gene would have no, would selection never touch? Any ideas? Okay. Probably not, because that's still making something. What parts of the genome don't make anything? What parts of the genome get ignored? Well, recessive still has an effect, but you're getting closer. Think about the spliceosome, if you remember that from 101. Yeah, the part of the chromosome that never gets used. What was that called? You got the expressed regions of genes, the introns. Who said that? Good job. The introns. If you have a mutation in the intron, it will never be expressed in the RNA. Oh, I'm sorry, it will never be expressed in the RNA, and so it will never be expressed in the protein. There's no way it's selected for. So it can change, and there's no problem. This type of evolution is known as survival of the luckiest, or non-Darwinian evolution. You have changes in allelic frequency simply because you have changes. They aren't actually selected for or against. It's not just the codon that's the issue. I'm sorry, it's not just the intron that's the issue. It's the third codon. Do you remember the genetic code? That's this box or the circle. You've got your first nucleotide, second nucleotide at the top, third nucleotide along the side. If you remember, the first nucleotide means a lot. Second nucleotide means a bit. Third nucleotide, if you look at this third row, if you have GGU, GGC, GGA, or GGG, you have glycine. It doesn't matter what else. You can change that third codon, and you will have no impact for the most part on the gene. I'm sorry, on the final protein. Here, GCU, GCC, GCA, GCG all make alanine. Here, GUU, GUC, GUA, GUG all make valine. It doesn't matter if you have a mutation at that third codon for the most part, because it's all of those codons encode for the same exact amino acid, which means they'll have the exact same chemical relationship to other amino acids in the protein. You won't actually change your polypeptide. You won't change your primary structure. You don't change your primary structure. Your secondary, tertiary, and quaternary structures don't change. So you have no functional change. Selection can't act on it. The only selective pressure you have here is genetic drift. All right, so that's enough about drift. With migration, you need to have gene flow from one population to the next. And as an organism moves from one population to the next, it takes its genes with it. Here we got a green population of beetles, brown population of beetles. The brown population of beetles, if one of them migrates over, suddenly introduces new genes to this population. Now, this population might start producing more brown beetles eventually. And then selection can start acting on that. Ultimately, the more migration you have, the lower the difference in allelic frequencies you have between two populations. So if you have, I don't know, lots of blue over here, very little red, and then over here you've got lots of red and very little blue, and you have a little bit of migration, you're going to see over here still a lot of blue, and over there a lot of red. But the more migration you have, the more even the allelic frequencies become between the two populations, until they become one population that transfers their DNA back and forth very, very easily and very readily. Non-random mating. This is like a type of selection where individuals choose their mates based upon their phenotypes. And assortative mating is a specialized part of non-random mating. With assortative mating, you might have homospecific crosses or heterospecific crosses. Homospecific crosses, like mating with like, leads to lots of organisms that are alike. If you have, I don't know, white dog and you mate them together, you expect white dogs. Does that make sense? Especially if you got those pure breed dogs that you expect one after the other. Like and like make more likes. Homospecific crosses. That leads to increased levels of homozygosity. The flip side, different to different will lead to increased heterozygosity. Those are called heterospecific crosses. If you take two organisms that are not really related to each other, cross them together, you have a lot more allelic diversity within the organism. More heterozygotes. Take a brown dog and a black dog, cross them together, you don't know what you're going to get. Even crazier, take, I don't know, a Chihuahua and a Great Dane and cross them together, who knows? Also, that's a tough one to do. So that's assortative mating. You could either go with the same, the homospecific crosses leading to increased homozygosity, or different, leading to heterozygosity. Inbreeding is going to occur when you do those homospecific crosses over and over again. You have individuals that are related to each other interbreeding. In the animal kingdom, this can be a problem, especially in managed species. If you've only got a few organisms left of a species, you've got to mate them together. Well, you don't have to, but you know, it's kind of, we like doing that sort of thing. And so any recessive alleles that that organism may have had are going to turn up more and more often in the offspring. Here we've got an affected mother. She's got some weird affected alleles and an unaffected father. They have some kids, and you end up with carriers, heterozygotes that are displaying normal in the first generation. If those kids mate, you're going to be much more likely to see a homozygous recessive phenotype pop up. Most of these homozygous conditions aren't going to, they're very rare. I know we keep doing these Mendelian crosses and we see 1 out of 4 chance, right? But in a population as a whole, they don't pop up that often. They're really, really rare, except when you start inbreeding. And then you see them over and over and over again. So if we have these snakes, and one snake gets a mutation, this is how inbreeding, this is actually how a lot of recessive conditions happen. You have 2 snakes in a population. One of the snakes has a mutation on one allele. That snake does not show an abnormal phenotype. It's got the other allele to make up for it. They have offspring. Well, now, 2 of those offspring are likely to have that recessive phenotype. They're also heterozygotes. And if you cross those 2 together, now you got a 1 in 4 chance of having an affected offspring. So by having these inbreeding relationships, you end up having a lot more homozygosity occurring. When the homozygotes have a lower fitness than the heterozygotes, you get inbreeding depression. So if homozygotes can't produce as many offspring, or don't produce viable offspring, suddenly you've decreased the fitness levels of that group, and that's sort of a way of preventing inbreeding from happening a lot. Now don't get me wrong, humans have been really big on inbreeding. Humans have inbred for generation after generation after generation. It's actually a fairly recent thing, a fairly recent cultural motif not to have inbreeding. In fact, Charles Darwin married his first cousin. I mean, that doesn't actually say much. Think about the royal family of England. Most of them have hemophilia. Why? Because they keep marrying their cousins. It's established. It's there. The pharaohs in Egypt. The pharaohs were considered to be gods. The royal family, they were gods. They couldn't mate with normal people. They could only mate with other gods who, you know, happen to be their brothers or sisters, or mom, or their dad. It was there. So it's there's been a lot of human inbreeding studies that happened. When a culture or group first starts inbreeding, they have to worry about these things called lethal alleles. We actually have a built-in system to stop inbreeding in our bodies. You have in you a ticking time bomb, a set of ticking time bombs. Luckily, it'll never go off in you. But if you had kids, and you have a lethal allele, and your mate matches your lethal alleles, your offspring won't be conceived. Well, they might be conceived, but they'll never make it to birth. In fact, a lot of those times when you guys hear about inbreeding and birth defects and things like that, what they're talking about are these lethal alleles kicking in. Lethal alleles exist within populations. They exist within all of us. In America, in this area right here where we are, we actually have a significant number of lethal alleles that hopefully stop inbreeding. Now, you guys have learned about evolution and selection. If you have these lethal alleles and you start inbreeding, what's going to happen to the offspring? They're not going to make it. What's going to happen to those alleles? The lethal alleles, well, they're homozygous, but the homozygous alleles, they get wiped out, so you're losing lethal alleles. One generation, lethal alleles are decreased. 2 generations, they're decreased further. 3 generations, they're pretty much gone. After several successful generations of inbreeding, you lose most of the lethal alleles. You can have cultural inbreeding at that point. I don't want to say okay. That's a moral, cultural thing. But biologically, it's no longer harmful. There are plenty of communities, plenty of cultures that still practice inbreeding as a way of passing material and passing property from one generation to the next. The daughter marrying the father's brother, things like that. You're passing, there are no more lethal alleles to deal with. It's one, and I know you guys, a lot of you guys are like, inbreeding, this is terrible. So we live in a pretty small area here. If any of you guys had to check around to see if you're related to anybody before you started dating them? No. Good. Accidentally went on a date with it. That happens. Good things to know, right? Yeah, those things happen. If you're from a small town, that occurs. And it actually occurs the closer you get to the Appalachian Mountains, the more likely it is to happen, because there's smaller and smaller, more confined populations. So that's societal inbreeding. Here's the thing, though, with societal inbreeding. One generation, just one generation of random mating, one generation of outbreeding, one generation of mating with somebody from somewhere else screws up the whole thing. You start the clock over again. They've brought in new lethal alleles. You have to go for another several generations before those lethal alleles are bred back out. So I guess the takeaway message is, if you're going to start inbreeding, don't stop, because then it's just going to mess up your family forever. Don't start inbreeding. That's what the other takeaway message. Just keep outbreeding. There's nobody good around here anyway. Just look elsewhere. In managed species, inbreeding becomes a major issue, because inbreeding depression can end up wiping out species entirely. Sometimes, when you've got such a small population that you're really worried about inbreeding depression, you'll actually bring in another species that's very, very closely related. I think it's the ibex and the goat. The ibex was a species of very, very, very large goats, and they were dying out. In fact, we know exactly when the very last one died, because we saw it happen. We didn't do it. It's okay. We actually were trying to protect. This is a great story. Okay, quick story. The last ibex. So we knew the ibex was on the verge of extinction, so we're trying to save it, and we mated it with a lot of goats. And we produced all these goat-ibex hybrids. Now, are those goats or are those ibex? What do you think? We don't know. They could be goats. They could be ibex. We have all these goat-ibex hybrids, and we had the last ibex. And there's the last ibex. I'm looking at it. Standing on a hill. And he's on this hill, and right above him is a tree. And he's looking majestically. I imagine the sun setting. I don't know if they have it on video, but they definitely have pictures. And there's a crack. And a limb on the tree fell, nailed the ibex, and killed it. And that was the end. That was the very last of the ibex. Died because a branch broke off and broke its neck. But there's a whole new project now. Now they've got these ibex goat offspring, right? They sequenced the ibex DNA. They know exactly what ibex DNA is supposed to look like, what the genome looks like. So now they're crossing those goats back, and they're in every generation. And each generation, they're picking the goat that has the highest amount of genetic material from the ibex. And they're saying, okay, that's closest. And they cross it to another one that has a different part of the ibex's genome. And they keep doing that over and over and over again. And they've gotten up to 95% ibex to bring it back. Isn't that neat? Yeah, managing extinct or dying species is really difficult. You have to first decide what a species is, and we'll get into that in the next lecture, actually. The red wolf and the gray wolf. You guys heard of the red wolf? The red wolf is an endangered wolf species. You can go to the museum, you see this red wolf. They've classified it as endangered, and they've tried to breed them in captivity and reintroduce them to the wild. Well, it turns out that the red wolf is very likely just a hybrid between a coyote and a gray wolf. So it may never have been its own species, and here we are just like, we need more of them! Put it out there. You got to figure out what the species is first, and then you can manage it. So in the next lecture, we're going to talk a whole lot about what is a species, and what does it mean to classify things? How do we classify things? Do you guys have any questions? Got some review questions for you if you're interested. If there are no questions, we'll just call it there for today.